Welcome, ladies and gentlemen. My name is Tristan Williams and I'm the Technical Director for the FinTech Group. Welcome to my presentation today on Room for Improvement, a presentation discussing the importance of responsible self-regulation for UVC device manufacturers. The UVC room disinfection sector has never been more important than it is right now. There is a driving need to collectively ensure that we do not mislead, confuse, or generate false expectations to our consumers. This is especially prevalent at the moment. Disinfection has always been an extremely important and continuous requirement, particularly in hospitals and healthcare, and COVID-19 has only served to emphasize this even further. We have gained new market sectors and a whole new generation of consumers. However, all of this can easily be unraveled by unscrupulous behavior in the marketplace. Think of this from a consumer's perspective. What we are generating at the moment for this new generation of consumers in our growing marketplace is loud, nonsensical noise. We are generating simply noise. There's a lot of headline figures, there's a lot of key metrics that are spoken about, and often spoken about falsely. Put yourself in the place of someone who is looking to choose a UVC disinfection system at the moment. How would you feel? What would you choose? What would you think is important to you? Especially considering the amount of noise that's coming out of the marketplace at the moment. This is an internally and externally recognized requirement. A recent white paper, which you can see referenced on the screen in front of you now, internally within this market sector, recognizes that in the absence of an established protocol and guidelines to validate commercial UVC disinfection products, numerous UV-based sterilization devices with unknown efficacies against the COVID-19 virus and lack of safety causes a major concern. And this concern also includes whether these products are yet at a stage to be used by these new consumers or amateur users. This is highly concerning, and this is really what I want to speak about today. But first, allow me to give you some context behind what I'm speaking about at the moment. Safety. Let me give you an example of what's happened with safety. For everyone that I'm speaking to today, safety is a key priority. It is one of our primary concerns, and we spend a huge amount of time engineering bespoke solutions and ensuring that we have the right hierarchy of operation to ensure that there is no adverse incidence of UVC exposure to any of our consumers or any of their clients. However, take for example the Cobb County School District install, where they installed overhead germicidal fixtures into the ceiling. And this cost a grand total of $768,634 across multiple sites. Now that's a lot of money, and they spent this money, but nonetheless, even with that amount of money that had been spent, there was a systemic timer malfunction which exposed pupils to germicidal UVC light. This resulted in serious adverse publicity, and you can see some examples of that serious adverse publicity on your screen right now. The downside with this adverse publicity was that the keyword was UVC light. And that affects all of us in this industry. That affects everybody, no matter how good or how poor a job you're doing at engineering your systems to be safe. The fact that UVC light was the keyword means that everybody in this industry has been damaged by this adverse publicity that's come about as a result of this poor quality installation. Put yourself in the consumer position. Again, this is part of the noise coming out from the industry. This is part of hearing what the media are reporting, hearing what public opinion is. How damaging is it to us, to a consumer who wants to use this technology, that then says, hang on a minute, I heard something on the news about that the other night, and that it isn't safe. The unfortunate side effect of the industry we're in at the moment is that we are all tarred with the same brush. Another key example is power. What do we define UVC power as? And this has been a really big issue that I've noticed creeping into the marketplace over the last year, specifically since the COVID-19 virus outbreak and global pandemic. Now, we all know in the UVC sector that the amount of UVC light that you generate with your system is absolutely critical for the dose calculations. It determines the time, it determines the efficacy. It lies at the very foundation of the industry that we all work in at the moment. However, I have seen many, and here's an example right now, companies claiming their electrical power consumption as their UV output. This is misleading. This is dangerous. And claims such as this will and have, I have already seen this happen, trickle down to tender specifications. This makes our lives as reputable UVC disinfection manufacturers even more difficult than they already are. How can you comply with a tender when it's asking for a minimum of, for example, 2,000 watts of UVC energy? You would need multiple systems across multiple radial arms or domestic circuits to be able to even power that, to get that amount of UV energy. So we have to be really careful with these kind of statements. They are damaging. Headline spin marketing such as this is confusing to consumers and damages this sector as a whole. Again, we are all tarred with the same brush. Efficacy is another key metric that I've seen recently abused with headline figures and claims and so on and so forth. Such as here, where you can see devices that claim to have 99.99% efficacy against all bacteria and viruses. For those of us that design, build, manufacture, and safely produce and test UVC devices, we know that that is a completely meaningless benchmark. Why? Because it's that log reduction, for example there we have 4 log reduction on which organism? Sporicidal, vegetative, COVID-19. Every single organism has a different dose, and those of us that design these systems live and work every single day with the Kowalski Index, which is a fantastic document that details the kill values for all of these particular organisms. And yet, still these claims exist, and it isn't just small companies that make these claims, but log reduction on which organism? But furthermore, on which material? Even if you've tested in a lab, which material was it on? Was it on a stainless steel plate? Was it on a petri dish? Was it on a textile or a fabric? These all change the way that the log reduction works and replicate or don't replicate real-world applications as they see fit. Which orientation or angle of incidence was this particular test done with? Remember, all this data, all of these log reductions that we see on a daily basis by various manufacturers making these claims, and again, it is big ones and it is small ones, it isn't just a particular type of manufacturer making these claims. All of this data is meaningless without the context. Unless you can understand how, what, where, and why it was tested with that particular organism on that particular material at that particular distance at that particular angle of incidence, then this data is entirely meaningless. Furthermore, time. Now there's no two ways about it. Time is important for our clients. Time is a key metric. We're talking about a device and a procedure and a process that is an adjunct to their existing cleaning processes. And time is money. So there is a driving need for customers to understand the time. And those of us that work with UVC systems understand that time is relative to dose. Intensity over time. That's how we calculate UVC dose. But there are very broad spectrum statements being made by a wide variety of manufacturers of UVC systems. For example here, this company here, which I've removed all identifying marks from in the interest of professionalism, are stating that their system is proven to destroy 99.99% of surface and airborne pathogens in a 1,000-foot square room in less than 7 minutes. They go on further to say that they achieve a hospital-grade efficacy. Now, I don't know their system particularly well, but I'm well versed in UVC, how it works, and the amount of power you need and duration that you need to disinfect a 1,000-foot square room to a hospital-grade level. Furthermore, if you're going to then state we do surface and airborne pathogens, then quoting a square area value is meaningless. Anytime you're quoting airborne as well, then you need to understand that that is a cubic volume, not just a square foot. That's a real issue when you're claiming you can disinfect airborne pathogens, because ceiling height makes an enormous difference to the cubic capacity of the room, of the treatment area, and has to be factored in. Talking square feet is not relevant when you're talking about airborne pathogens. It needs to be a cubic volume. If you are going to be making bold statements such as you can disinfect an area to a hospital grade level within 7 minutes, then that should be backed up by suitable evidence, or at least specifying the circumstances of that particular test. Because the dosage is time, but it is relative to room size, your, in this case, cubic volume, because we've also discussed that the statement is made about airborne pathogens. The contents in the room, because contents generate shadows. Rooms are very often considered just simply to be empty cubes, but in a lot of applications, it's very rare to find that a room is completely void of furniture or void of any kind of contents. The shape of the room makes an enormous difference because it changes the angle of incidence of the light and therefore the efficacy of the light. The location of your device should be stated. Was it central? Was it offset? Because that is how we deal with the radiant inverse square law. The light, for most UVC systems that are static, comes out in a radius, and therefore is directly and easily and tangibly subject to the radiant inverse square law. And what was your target organism? What were you looking to kill within 7 minutes? What was the dose that you were trying to achieve a 4 log reduction of that particular organism? All these factors need to be considered before making statements such as the below. Otherwise, again, the UVC sector is going to be damaged by these misleading comments. Let's flip that coin around a little bit. I want you to put yourself in the position of a consumer. I want you to be looking for a UVC system, and you've just read the statement below. What does that mean to you? Now, if you read that statement and it told you it was on a particular virus that you were looking to eradicate, in this case, for example, let's consider COVID-19, that the ceiling was 2.7 meters high, that there was 3 tables and 3 chairs in the room, that it was carpeted or it was vinyl flooring, that the room was ventilated, it would give you a really good idea of whether that system is suitable and whether the evidence backs up the performance. Headlines are not answers. This also brings me on to an even more concerning issue, which is that certain UVC suppliers are claiming false approvals. And this image here illustrates that perfectly. Now, this image is of a decal that was applied to a system, and they were claiming that the system was FDA approved. Not only in their product images, but in their product videos and in their product literature. The FDA themselves state that the logo should not be applied to a product. And furthermore, that you should not say the product is approved unless it's a Class 3 device. If you're using a 510K, then you use the terminology of cleared. If you're using the de novo pathway, then you would say granted. And that is highly concerning. What's even more concerning is that to a consumer, this looks fantastic. FDA approval. Superb. Excellent. That must mean it's been tested, validated, and released through the FDA pathways. So what does the future hold for us here in the UVC sector? What's the purpose of my presentation today? It certainly isn't to besmirch any other manufacturers. That's not my intention. My intention is to engender a conversation, to empower people to begin to look at this collectively as an industry. How can we self-regulate better? I don't have the answers. I simply would like to start the conversation. I'm a believer in this technology, a real believer, a convert. I am an advocate. We are all advocates for this technology. But what I'd really like to close on is I want everyone that's watching this presentation today to not forget the reason we are here. We are here to prevent infections and protect the public health. We have a genuinely important role to play in antimicrobial stewardship. We must take action to ensure that we are all collectively doing the right things for the right reasons. Thank you very much for your attention today.